Hey guys, welcome to chapter 12.3. We are talking about the cell cycle and how it relates to cancer. So let's get started. So in the 1970s, scientists wanted to know if what regulated the cell. Is it internal cues or is it signals or is it external signals? And so one scientist put in a petri dish a cell that was in S phase, and in another petri dish, he put a cell that was in M phase. And then he put cells that were in G phase with each of these cells, the one with S phase, and he put a cell that was in G1 phase with the M phase. And what he saw was that the one that was in G1 phase that was put in with the S phase, it immediately went into DNA replication. So the G1 took on the same phase that the S phase, that the S1 was in. And the G1 cell that was put with the one that was in M phase immediately started preparing for mitosis. So this showed the scientists that the cues or the signals for regulating the cell cycle are internal. They are happening inside. And so there is a cell cycle control system. And that is what we're going to talk about right now, is the cell cycle control system. And as you know, the cell cycle consists of interphase. Interphase is 90% right here, 90% of the cell cycle, and it consists of G1, S, and G2. So we have G1, S, which is DNA replication, and G2. And at the end of G1, at the end of G2, and at the end of the M phase, there are things called checkpoints. So there's a checkpoint before the cell goes into DNA replication. There's a checkpoint before the cell goes into cell division. There's a checkpoint before the cell divides into two. And what happens is the cell cycle just sort of slows down, and it checks to make sure that everything is working correctly. And then it registers external signals to continue moving the cell along. But it makes sure that all the crucial processes, these checkpoints, make sure that all of these crucial processes have been completed and that are done accurately, so that we're not replicating a cell or replicating chromosomes that were incorrectly placed or what have you. So these checkpoints make sure that the cell is in good working condition before we move on to the next phase. For some cells, the most important checkpoint or the most important phase is going to be the G1 phase. So when cells go through G1, most of them are going to go through G1, and if they do, they're probably going through S phase and G2 phase and through mitosis, and they're probably going to replicate. However, there are some cells like neurons, like liver cells, muscle cells, some muscle cells, not all muscle cells, that are going to stay in this. It's not going to pass through. So once it reaches that G1 checkpoint, it's actually going to trigger it to go into this G0 or G0 phase. And it's going to stay there for the rest of its cell life because some cells can't shut down for the time that it takes to replicate. So your brain cells can't shut down for an hour or however long it's going to take for that specific cell to replicate. And so it's going to remain in an active state in this resting G0 phase, and it's just going to stay there. And so those cells are going to stay like that, and then the rest of the cells are going to continue going on through all of the rest of the cell cycle. So what brings a cell through these checkpoints? Well, the key players in this process are going to be protein kinases and cyclins. And kinases, as you know, are enzymes that activate or deactivate a protein by phosphorylating them. They add a phosphate group, and they're going to turn proteins on. And so in this scenario, not only do we have kinases turning proteins on, but we now have something that's going to need to turn the kinase on. We have cyclins. Cyclins are going to attach to kinases, and then those cyclin-dependent kinases are then going to phosphorylate or add phosphate groups. So cyclin-dependent kinases are kinases that require cyclin. And the kinase concentrations are going to remain steady. So let's say that let's use an imaginary scenario where in a cell there's, let's just say a unitless number. There's a hundred kinases, and the kinases are going to stay at that same level. There's going to be 100 kinases or cyclin-dependent kinases in the cell at all times. What does cycle? What does go up and down according to where the cell is in its cell cycle is the cyclins. This is why we call them cyclins, because they increase and they decrease depending on where the cell is in its cycle. And so the kinases remain constant, but they're not always working. They only work when the cyclins are present. And so when the cyclins, we can look at this graph here, when the cyclins increase, the cyclin concentration is purple. When the cyclins increase, then the cyclin-dependent kinase will then do its job. We call the cyclin-dependent kinase and the cyclin together, we call that a cyclin-dependent kinase complex. So when we have a cyclin-dependent kinase complex, when we have both the cyclin and the kinase together, that is when that kinase is going to be able to do a job. However, well, let's look at an example. So there is a cyclin-dependent kinase complex known as MPF, the M phase promoting factor. So this is the molecule that's going to move the cell forward into M phase. And so if we once again look at this graph, we know that we're going to have a steady number of kinase in the cell at all times, through M, through G1, through S, through G2, back through M, G1, S, G2. However, we can see that the cyclins increase. So here we have in pink the cyclins increase. And so when cyclins increase, we can see here that the MPF, or the cyclin-dependent kinase complex, is going to increase. And so an example of what these MPFs are going to do is they're going to phosphorylate proteins, which is going to initiate mitosis. So, for example, the MPF is going to phosphorylate a protein near the nuclear envelope, which is going to start to break it down so that the chromosomes can start to move around out of the nuclear envelope and move towards the metaphase plate. So once mitosis is complete and the proteins are no longer needed, so once we get through that M phase and we no longer need the MPFs or the cyclin-dependent kinase complex, then the CDKs or the cyclin is going to be degraded. It's going to detach, it's going to break down, and so then we just have kinase in there, and they're not going and working on proteins because there's no cyclin attached to it. We use cyclins and kinases to control the cell cycle and to signal through the cell cycle. However, there are other mechanisms that are used during the cell cycle. An example of this are like during the M phase. M phase will not continue past anaphase until every kinetochore is attached to a spindle microtubule. So once this has happened, the regulatory protein complexes can become activated, and then they release the enzyme separase, which we know is going to split the cohesions between the sister chromatids and then allow the sister chromatids to migrate toward the pole. But that's not going to happen until these proteins are activated. So there are many different mechanisms that control the pace of the cell cycle, and many of these are still currently being researched. So we've talked about some internal signals, but there are also external signals. Growth factor is an example of one of these. And so growth factor is a protein that is released to stimulate cells to divide. So platelet-derived growth factor, or PDGF, is found in platelets in the blood. And so platelets contain a clotting factor. So if you get cut, your cells are going to release PDGF. They're going to They're going to release clotting factor. That's going to help you make a scab, so you stop bleeding. But then it's also going to deliver this PDGF. And so what happens is it tells this PDGF is going to tell the cells to pass through G1, and that's going to signal them to divide, and they're going to keep dividing until they reach another signal to tell them to stop dividing. And so this is going to help to cover for the heal to wound. It's saying keep dividing so that we can stop bleeding. And so cells will continue to divide until they are signaled by another external signal, and this is another cell. So when cells come in contact with another cell, when they are surrounded by cells on all sides, they will stop dividing. This is called density-dependent inhibition. So if we have a Petri dish, and on the bottom of the Petri dish we have a few cells, those cells are going to continue to divide until they have all reached each other. And then once the whole bottom of the Petri dish is covered, then they will stop dividing. If you were to pull some of those cells out, then they're going to divide and fill in that space. Another external signal is called anchorage dependence. This means that in order for cells to divide, they need to be anchored to a substratum. That's so that you don't have cells just dividing willy-nilly through anywhere in the body. They need to be attached to something else. And if there is no attachment, they shouldn't be dividing. And so when we lose control of these controls, we have cancer. So when we can't control the rate of the cell cycle, when something happens that it speeds out of control or we can't stop it, this is cancer. Cancer is uncontrolled cell growth. And so these often occur because of genetic mutations that alter the way proteins work and change the transduction pathway of the cell cycle. And so sometimes a cell will divide in the absence of growth factor. Sometimes it will produce its own growth factor. And so therefore it's going to be replicating and growing into a tumor. So cancer cells can have many odd traits about them. And if and when they stop dividing, they can do so at any point in the cell cycle. So they can stop at G1, they can stop at M, they can stop in the middle of replication. It's not just at checkpoints. So they're kind of deranged almost. And cells that have undergone a process called transformation, they can continue to divide forever. What this means is that they have overcome the sort of natural stop that we have in our cells. So usually our chromosomes can only be replicated about 20 to 50 times. That's because on our chromosomes, we have these caps and poly A tails. We have these because every time your chromosomes are replicated, they lose a little bit of the ends. And so once those ends and those caps start to degrade and sort of disappear, the cell is going to, or that line is going to end. Those chromosomes can't be copied anymore. However, cells that have undergone transformation have figured out how to copy the whole chromosome, and so they can divide endlessly. And this is right here is a YouTube video for HeLa cells, or Henrietta Lacks. It's a great book, and it's a great story about a cell line coming from a woman who didn't know that she was giving up her cells. So very interesting story. So the loss of cell cycle controls in cancer, they are going to occur if apoptosis doesn't occur. Apoptosis doesn't occur, or a cell evades the immune response of being destroyed, then tumors, it's going to continue to grow. They're going to replicate, and it's going to cause tumors. Now, there are different types of tumors. There are benign tumors, which are going to grow, and then they're going to stop. They're not deranged enough. They're not so mutated that that they figured out how to spread and to grow into different parts of the body. So they're going to grow, it's going to replicate, and you actually have thousands of benign tumors all over your body. They're not so mutated. There's not mutations in there to allow them to continue to grow. And so malignant tumors, though, have genetic and cellular modifications that allow them to spread, allow them to go to other tissue, to impair brain function or lung function, organ function. And so these cells have many unusual numbers of chromosomes, or they can have altered metabolisms. They can change the cell's surface, which allows them to divide regardless of their attachment to a substratum. And so many cells can secrete a signaling molecule, which is going to signal or call for the growth of blood vessels. This is called angiogenesis. So in this picture, we have a tumor, and if it doesn't have blood, it's not going to continue to grow because it doesn't have a blood supply to it. And so in order for cancers to grow, it needs to have blood, it needs to have food, nutrition. And so the tumor can send out the signaling molecules to call for the growth of new blood vessels. This is called angiogenesis, and it allows the cancer to enter the bloodstream. And once the cancer enters the bloodstream, this is called metastasis. The tumor has metastasized. So there are many new treatments for cancer out there, but most commonly we use chemotherapy. Chemotherapy, what it does is it stops rapidly dividing cells. And one example of this is Taxol. Taxol freezes the mitotic spindle. And so when the chromosomes begin to migrate to the opposite sides of the cell, the spindles don't depolymerize. And so this leads to cell destruction. So you've got destruction of all of your newly replicating cells, which means that the lining of your stomach and of your small intestines, your hair follicles, your immune system, all of these systems that require cell growth or new cells is going to not have that. And so it leads to people having hair loss, susceptibility to infection because there's no new white blood cells being created, and nausea because the lining of your intestines is not regenerating. So it is sometimes thought to be more difficult than the cancer itself. But these are the notes, and please let me know if you have any questions.